welcome to the Engineering Biology Research Consortium Education Module on Synthetic Biology and Machine Learning. I'm Mary Dunlop. I'm a professor of biomedical engineering at Boston University. In this series, we'll be talking with researchers who work at the interface of synthetic biology and machine learning about projects in their group and ideas for how to learn more about this area. So we're joined today by three different people. So the first is Neythen Treloar, a postdoctoral scholar in computational synthetic biology at the University College London. Welcome, Neythen.
Thank you.
Next, we also have Brian Ingalls, a professor of applied mathematics at the University of Waterloo.
Great to be here.
And Chris Barnes, a professor of cell and developmental biology at University College London.
Hi, thanks.
Great, so welcome to all three of you. And in this discussion, we're going to be talking about a paper that this group published that uses reinforcement learning coupled with neural networks to control the composition of co-cultures within bioreactors. Neythen led the study, and it was jointly published, jointly supervised by Brian and Chris, and the article appeared in PLOS Computational Biology in 2020. All right, so we're going to get to the modeling parts of the work in a little bit, but could you start by telling us about the overall goal of the study?
Yeah, so I have a couple of slides about this. So obviously, if we think of just to introduce kind of industrial bioprocessing, we know that we can take a bacteria and we can kind of engineer that bacteria. So we can put sort of artificial genes in there. So for example, if we wanted to produce human insulin, we can engineer a bacteria to do that. And then we can put that bacteria in a bioreactor and kind of, grow it so that it produces insulin. And that's a very useful thing to be able to do. But then kind of as we go forwards in synthetic biology, we will probably become a lot more interested in using more complex kind of communities that are composed of more than one strain of bacteria or even different, completely different species, so like E. coli and yeast. And there's a few things that motivate this. So there's a few papers which show that this could be kind of more efficient for a few different functions. And also, if we're just looking at one bacteria, we are kind of limited by the metabolic capability of that cell. So we're limited basically in the complexity of the reaction pathway we can put in there, if that makes sense. Yeah, but the problem with this is basically, increasing the complexity of a community basically makes it kind of harder to control. And one of the reasons for this is competitive exclusion. So obviously if one of them grows faster than the other and they share like a common resource or something like that, you'll basically get one strain outcompeting the other, which is, not what you want. So basically we were investigating reinforcement learning as kind of a possible solution to this as like a new sort of control method in these situations.
Can you tell us a little bit more about what is reinforcement learning? And also you talk in the paper about this approach being model-free. Can you describe what you mean by that and why it's important?
Yeah, sure. So I have this like kind of slide which kind of breaks machine learning down into kind of three broad categories. So it's kind of unsupervised learning, and it's all about like finding like structure in data. So that would be like, clustering different data points or trying to find like the most important factors in the data set. Then you have like classical supervised learning, which could be like classification. So if you're trying to classify images, you'd have a set of images and you'd have labels like this is a dog, this is a cat, this is a house, and you try and like classify them. And then you have kind of reinforcement learning, which I might be slightly biased, but I think that's kind of the most interesting one. So the idea behind reinforcement learning is we're trying to teach an agent optimal behavior. So we have an agent that lives in an environment. And you can kind of think of the agent like a dog, right? So if you're trying to train a dog, what do? You might be trying to train it to sit. So you tell the dog to sit, and the dog's going to hear that command, right? So that would be kind of the dog observing the state of its environment. And then based on what it hears, the dog might do an action. So it might sit, in which case that's a good kind of action, right? and it's going to get a positive reward. So you're going to give the dog a treat, or the dog might just like ignore you or run away or something like that. So that's obviously a bad action, and it's going to get it's not going to get any reward for doing that. And then what the dog will do is alter its behavior to basically receive as many treats as possible. That's kind of what a reinforcement learning agent does. So it kind of lives in an environment and it observes the state of its environment. Then it will take an action, and depending on the result of that action, it will get a reward. And what it tries to do is basically tries to maximize its total reward that it receives over time. So I hope that makes sense. And then so a lot of the kind of research in this has been done in kind of video and board games. So a big deal was kind of superhuman performance in Go, which is a really hard problem. But also we have kind of more recent, more practical applications for like computer chip design, which is very cool, and kind of like nuclear fusion control also. And kind of the reason we say this is model free is because it learns based on experience. So it doesn't need to kind of understand the mechanism of the environment that it's interacting with. It just learns by observing these states and rewards and the effect of its actions, if that makes sense.
Right. So just to clarify that point a little bit, so that like alternatively, you could have a model that you write down that's like, say, ordinary differential equations or some other system that describes it, but you don't have to know anything about those details in this kind of scenario. But you do still have a model in the sense that you have like a machine learning model that you're working with. Is that correct?
Yeah, exactly. So you'd have like a machine learning model, as in the agent has. We'll talk about it more in depth, but yeah, the agent will have some kind of model that tells it which actions are good in which environmental states. But yeah, you don't need like the set of ODEs that describe the environment beforehand or anything like that.
Okay. Mm-hmm. A little bit more about the machine learning model itself. What are the inputs and outputs to the model, and how do you represent them numerically?
Yeah, so it might just be best to kind of explain the system setup, so just to give a bit of context. So we have our kind of training loop, and so we're obviously kind of looking at controlling these communities of bacteria. So we basically we model this system in which you have 2 strains of E. coli growing in a chemostat. So we have constant through flow, so constant input inflow and outflow. And then we assume we can measure the population levels. So you can do that with fluorescence, for example. And that basically makes up the agent's state. So the agent is observing the population levels. And then the actions that the agent takes is to control these nutrients into the bioreactor. So we model the situation in which both of these bacterial strains are auxotrophs, which means they're dependent on a certain nutrient. And the agent basically controls those nutrients to control each population. So there's kind of sort of a diagram up here. So we have our 2 strains. They each have an independent nutrient, but then they both compete over the carbon source. So we have competition there as well. And then kind of an important thing is that the agent is acting as a bang-bang controller, which this means that for each nutrient, it can either turn that nutrient on or it can turn it off at each kind of time point. So if you think about if you have 2 nutrients, each of which can be on and off, then we're going to have 4 different possible combinations, because we can have off, off, on, on, and on, if that makes sense. And that kind of has implications for the model.
The fitted Q-learning approach that you use?
So kind of the main idea behind Q-learning is we do this kind of trial and error interaction with the environment. So we gather kind of experience, and then we use this experience to learn a value function. So what a value function tells you is if you give it kind of a state action pair, it will tell you the kind of estimated reward you can expect to get in the future, if that makes sense. So that's like kind of how good the state action pair is, because obviously a state action pair with a higher value is going to lead you to higher rewards in the future, if that makes sense. So then after you kind of learn this value function, you can, once you have like a good value function, the agent will, if it's in this environmental state, it will look at its value function and it can pick the action that has the highest predicted value, and that should give it the most reward, if that makes sense.
Okay.
Yeah, and then, so just to have a look at what the actual model looks like. So we're using a neural network. So the input is basically the state. So that's the two different population levels. Okay, so N1 and N2. And these are kind of normalized to be between 0 and 1. And then the output, so if you remember, we had 4 different actions that we can take from the bang bang control. And then the output will be the predicted value of each action. So what this tells you is like look, I've measured the state of my environment and I'm in this state. These are my population levels. And then the output, the Q function will basically tell you, okay, well, these are the predicted values of each of these actions. And you can see that from these example values, this one here would be the best action to take. So given all the experience that I've had so far, I think action number 3 is the best one.
Here is this the actual network. It's got 2 hidden layers.
It has 2 hidden layers, if I remember correctly, but it has 20, I believe, neurons in each layer. So it's not like a huge neural network. It's kind of relatively quite small.
Okay. How did you end up settling on the number of layers and the number of neurons in each? Was that like an iterative testing process, or how did you come up with that design?
Not really, to be honest. So I got the number of layers and the size of the layers from just from another paper that had implemented fitted Q learning, and it kind of worked. So I just kind of left it at that.
Fair enough. Yeah. Could you just briefly explain the difference between deep reinforcement learning and traditional reinforcement learning approaches, in the context of what you're showing on this slide?
Yeah, so deep reinforcement learning just means we're using a deep neural network. So that's like a neural network with more than one hidden layer. So we're using the neural network, to learn this Q function, but you can use kind of any kind of sort of function approximation, or kind of a common simple example is to just have a table. So if you have a discrete state space and a discrete action space, you can just have a table that, so for every state action combination, you just learn just a number, if that makes sense. So you can do tabular Q learning, which is quite common. Or you can have, you can do any kind of other approximation, like a linear approximation and things like that. Yeah, so basically we showed that so the first thing we wanted to show was that we could basically control the system. So we did that by setting kind of target population levels for each of the bacteria. So you can see in here, these are these kind of green lines. So this is a result of an experiment. So a 24-hour experiment after the end of training, and you can see that we've kept the populations close to the targets. And then so what we do in reinforcement learning is you often break your training up into episodes. So in this case, each of our episodes is one 24-hour experiment. And then after 24 hours, the episode finishes. Or if one of our bacteria basically goes extinct, so its population level gets too low, then the episode also finishes. And so what we have here is kind of how the agent learns over these episodes. So we have 30 episodes, and you can see that our reward increases as the agent learns. And then there's kind of another important thing is that you got to think of this kind of explore, exploit trade-off. As you can imagine, at the beginning before you've trained your agent, it doesn't know anything, right? So it's not going to want to exploit the knowledge it already has. It's going to want to take exploratory actions. This is captured by this explore rate here. So at the beginning of training, the explore rate is 1. And this means with probability 1, it's just going to take a random action. Okay, so it can kind of gather more experience. And then as our episodes progress, that explore rate kind of decays, and then by the end, the explore rate is 0. So it doesn't take any random actions at the end, and hopefully by that time you've learned good behavior. Just kind of one other cool result, which kind of goes into the whole like model free aspect, is we altered our system slightly. So we added these kind of intermediate compounds that are produced by each strain of bacteria. And then we have this sort of a kind of like dummy reaction where we're producing this product using this ratio of the compounds. And this time we change our reward function so that our reward is just the rate of product production. Okay, so this would be something you might want to implement realistically. And because this is quite simple, we know that the optimal population ratio should be 2 to 1 because of this this ratio here. And if we look at kind of the experiment kind of that happened after the agent was trained, and you look at the stabilized population ratios, those are like exactly 2 to 1. So that's good. And then sort of the other thing that we need to show is that all of our carbon that's coming in is being used up to produce this product. So the internal carbon in the reactor should be 0. And then this is kind of the carbon across our 24 hours, the level of carbon across our 24 hours for 10 repeats. And we can see that, we're using all of our carbon. So this is kind of good. It shows that just by observing this product output, we can basically drive the system towards the optimal state for maximizing the product output, if that makes sense. And we don't need to have any knowledge of how this all works, which is kind of an advantage of model free.
In the paper, you talk a bunch about this concept of the inter-sample period. Could you explain that a little bit, and why reinforcement learning tends to work well for longer inter-sample periods?
So the inter-sample period is so we have our experiment, which is obviously in continuous time. So we basically break that down into a set of time steps. And the inter-sample period is the period between those time steps. So it's the period between observing a state, taking an action, and then observing the next state and taking the next action. So reinforcement learning, you want your system to be Markovian, which means that, so the agent observes a state of the environment, right? And you want all of the relevant information to be encoded in that state, if that makes sense. So the problem is with the system, we have some hidden variables. So we only measure the population levels. We can't measure the kind of internal concentrations of these nutrients, if that makes sense. So it's kind of actually it's like a non-Markovian system because those nutrient levels are kind of relevant to the decision making process. But by increasing the inter-sample period, the system kind of becomes slightly more Markovian, or it looks Markovian. And an intuitive way to think about this is basically if the agent puts some nutrients in, you have to let the system run for enough time for that to have an effect.
So one of the things that I really like about this study is that you even though it's all simulations, you really appreciate that this is a space where it's just not feasible to generate incredibly large amounts of data in a realistic lab setting. And you show using simulations that this approach should be tractable in a realistic experimental setting. What adaptations or special considerations do you think this would be necessary for this to actually work in the lab?
So this is kind of something we thought of from the beginning. So the reason we chose fitted queue learning is because it's sort of more data efficient than kind of the more standard methods you'd use. So kind of even in these, we only use 30 episodes, where each episode is 24 hours. So that's already a really small amount of data compared to previous, other reinforcement learning work. But it's probably feasible to generate that amount of data. But obviously, if we can in a lab setting, but obviously if we can reduce it further, that would be even better. So what I looked at was if we assume that we can just have 24 hours worth of data, but we can run a number of bioreactors in parallel, like, how many bioreactors would we need to be running for 24 hours to get a good, to learn good behavior? So in this case, we just have one episode, which is one 24-hour experiment, but we're interacting with 5 bioreactors simultaneously, and kind of making independent decisions.
Okay, just to make sure I understand this, so that in the previous results that you presented, you were doing multiple sequential episodes where it was one bioreactor run for a 24-hour period. That was one episode, and then you would do that's another bioreactor, it could be the same one, for another 24 hours, and that was a second episode. And in this case, you're considering 5 running in parallel for a 24-hour period, and that's it. Is that right?
Yeah, exactly. Yeah. We interact with all these 5 bioreactors in parallel and make kind of independent decisions, and gather all the experience from them. It turns out that in this 24 hours, you can learn kind of how to control the system. So if we maybe it's better to look at this first. So again, we have these kind of target population levels, and we start the populations at the targets. And you can see that at the beginning of the 24 hours, kind of the populations just drift, right? But that's because so this is one of the 5 bioreactors, just to clarify. And this is because the agent's just, it's exploring. So we've got the explore rate here over our 24 hours again. So the agent's exploring, so, the populations are just going to drift randomly as it undertakes random actions. And then you can see that as the time goes on, the agent learns, and we kind of get our population levels down to our targets. And this is kind of reflected here as well. So this is the reward. So the reward is basically how close we are to the target. So if you're close, you get a higher reward. And if you're far away, you get a low reward. So you can see we start at relatively high reward because we start it in the right place. But then as it takes independent exploratory actions on each of our bioreactors, the reward drops, and we also get really high variance, right? Because it's taking, it's doing independent random actions. And then as it learns, the variance drops and our reward basically increases. So this is like really cool because we basically have 5 bioreactors which we could run in parallel for 24 hours. So we could do this experiment in the lab.
All right, so the study uses purely simulation data, but can you talk about any additional considerations you anticipate when this approach is deployed in the lab?
So I have kind of a list of 3 things. So Chris and Brian can obviously jump in. But so we, working with these tabletop bioreactors. So basically, we've been having to calibrate the sensors and the pumps because they're all kind of slightly different. So if you have 5 of them, each of the sensors will be slightly different. Each of the pumps will be slightly different. So that's kind of a slight issue. Then you have there's quite a lot of noise in the measurements, and then there's also kind of more delay on the actions, which could potentially be maybe a problem for kind of the reasons we talked about before, where we might have to increase the intersample period. But the kind of cool thing is that, especially with the kind of calibrations of the sensors and the pumps, it would be cool to investigate if kind of reinforcement learning could kind of learn to take into account the differences in the different sensors and the different pumps in its behavior. So, it could learn that bioreactor 1 has maybe, its sensor is off by this much, so it needs to adjust its kind of behavior to account for that, and things like that. And then there's also kind of ways we can get around this kind of delay thing, which makes it more difficult to look sort of Markovian. So there's ways around these, or reinforcement learning might even help with some of these problems, I think.
Yeah, I can just add that well, a lot of these tabletop bioreactors are designed to bidostat mode, which is actually an easier problem than chemostat in terms of the hardware. So I think the challenge is getting the chemostat mode working to a accurate enough level that we can control it predictably. But it's ongoing work in the lab at the moment.
So, Neythen, I have kind of a control background, so I'm kind of curious to hear how the reinforcement learning approach compares to like other more traditional methods. And I know in the paper you did a comparison to some proportional integral control. Can you talk a little bit about those results?
Yeah. So yeah, it was proportional integral control, not PID, as it says up there. But yeah, so basically we just kind of investigated the effect of the intersample period on both of these control methods. So to get kind of the PIDs, I basically used like MATLAB's automated like PI tuning toolbox as much as possible so I can be accused of basically just making a bad PI, if that makes sense. Yeah, so what we have here is basically changing the intersample period. And then what we see is that at small intersample periods, the PI, kind of, you can see it does better at 5. But then as we increase the intersample period, the PI kind of gets much worse, whereas the reinforcement learning kind of learns to sort of deal with the increased sampling period much better. I was just going to say these are the population levels at 5 minutes for the reinforcement learning and the PI control, and then this is them at 60 minutes, and you can see kind of the PI control is kind of much worse at 60, but at 5 they're very comparable.
Okay, and the 5 and 60 that you just mentioned, those are the different inter-sample period.
Yeah.
Values, right?
So basically at the extremes of the ones we tested. Yeah.
Yeah, so maybe I just add on to this that this is an important piece, because the reinforcement learning is really there are already solutions from feedback control theory, as you know, Mary, to these sorts of problems. And so really what we're trying to highlight, I think, is how this approach is complementary to those existing approaches, and how certain features might be more or less amenable to certain systems, given these rather different approaches to ultimately the same.
Yeah, that's cool. And I like because you guys have really taken practical constraints into consideration here. Like, you probably can't sample like infinitely quickly in the way that you might benefit from to get really accurate PI control. And so being able to extend that has practical experimental like implications. Yeah. For this example, like, are you still envisioning that you have the strains tagged with fluorescent proteins, or here you're just measuring the output that you're interested in?
So we still have to, in this example, measure the populations to make up the agent's state. You might be able to relax that, but then the only thing you'd be measuring is the product output, which, we didn't try that. Maybe it would work, maybe it wouldn't. I'm not sure. But yeah, in this example, we still have to measure the population levels.
Since the PLUS computational biology paper came out, I know you guys have been working on other applications and have a new preprint that's on bioRxiv where you're looking at other applications, including optimal experimental design. Could you tell us a little bit about some of the ideas behind that work?
Yeah, so we have kind of a very high-level overview. I didn't want to go into like too much detail, but yeah, we're kind of using sort of a similar method, so a similar reinforcement learning method to do optimal experimental design. So kind of the problem here is like given sort of a set amount of experimental effort. So that could be like we want to do a characterization experiment. The last in the paper is 20 hours. And we choose different experimental inputs every 2 hours, like something like that. And then the problem is the thing you want to do is you want to design those experimental inputs to basically gain as much information about the system as possible. So the kind of situation we looked at is if you have an ODE model of the system and you want to parameterize that model. So in that situation, what you want to do is you want to reduce the uncertainty in the parameters as much as possible. So we have kind of like two examples of a poorly designed and a well-designed experiment. So these are just like hypothetical examples, just to illustrate it. So there's kind of this metric called the Fisher information, and an experiment that gives you low Fisher information will lead to high uncertainty in the resulting parameter estimates, whereas an experiment that gives you high Fisher information will result in lower uncertainty. So the idea is that now we train a reinforcement learning agent, but the reward is basically this Fisher information here. So it's learning to maximize the Fisher information across a whole experiment, and that will give you less uncertainty in your parameter estimates of the system. So it's very similar setup to before. So it's on, again, like a chemostat system where sorry, we should say state, but the state that the agent observes is basically like population levels. The reward now has changed to be this Fisher information. And then the action it takes is the experimental inputs, so it's nutrients again. And then the idea is that we can do all of this training on the model, so the model of the ODEs, and then we can basically take that agent and we can apply it to a real system, and it can kind of do the experiment for us. There's kind of like a circularity here in that. The best experiment is dependent on the parameterization of the system. But obviously, we can't assume that we know the parameterization beforehand, or there wouldn't be any point in doing a parameter inference experiment. So it's kind of like a few ways around this without reinforcement learning, but we're kind of looking at reinforcement learning as like an alternate way around this. So how we can kind of do this is we can basically have a distribution over parameters. So in this case, we just have like a broad uniform distribution. So we're assuming like not much prior knowledge. We basically know kind of they're within this broad range. And then we train our agent as normal. But every episode, we take a different parameterization from this distribution. That makes sense. And then the idea is because like we don't know where in this distribution our real parameters are going to end up being. So the idea is that wherever we are in this distribution, we can do an optimal experiment. And we do show that basically this works. So what the agent will do is for different parameter samples, it will adapt its experiment depending on where it happens to be in that distribution, which means that kind of wherever you are in that distribution, you're going to get a near optimal experiment at the end.
So maybe I could jump in. Yeah. So again, if you compare to traditional approaches, something like an adaptive controller would be suited to solving the same sort of problem. And again, that's the idea of sort of finding complementary approaches and determining when something's going to be more useful or more tractable to a certain group in certain ways. And so one thing here also in terms of this opportunity to approach things as model free as opposed to sort of completely model based is this is sort of this is kind of taking the best of both approaches in the sense that you're taking advantage of your knowledge of the mechanism to put together a model. In this case, the training is done sort of over a collection of models with you're presuming the structure is sound, but you don't have a good idea of the parameterization, and then you're training over that sort of model space, and then the idea is you could deploy that controller over the real system, and if that's somehow been captured within that model space you've explored, then that knowledge of the model structure has been useful for that training exercise, and then you can deploy this in a model-free way once you're actually in the lab.
From a machine learning aspect, what I like about this is that you're combining the black box approach or model-free approach with a mathematical model, a mechanistic model. So you're kind of following on from what Brian says, but you're kind of combining the best of both worlds. And ultimately you train a mechanistic model, but you use the agent to do that. And I think this kind of interpretable AI is really important for use in biology.